Hey everyone, welcome to another episode of Genetics. Last time what we talked about was gene linkage. Today we're going to go a little bit further in depth into it and figure out how we can determine if two genes are particularly linked. So the way how we're going to do this is we're going to look for the recombination frequency, but we're also going to use something called map units and centimorgans as well. So the map units or centimorgans is essentially a unit that measures the recombination frequency between two different genes, but it doesn't actually measure the actual distance between two different genes. So it doesn't actually measure the distance, it just measures the recombination frequency. And all it is to calculate the map unit, or sometimes it's also called centimorgans as well. It's basically a recombination frequency times 100, and then that'll give you, the map unit or centimorgan between two different genes. You can also go in reverse direction as well. To figure out the recombination frequency from centimorgans or map units, it's pretty simple. To figure that out, you would get something like this. So map units or centimorgans divided by 100. To figure out the recombination frequency. And typically the way how you're going to see these written is the entire collection of genes will be written as on a linkage map. So a linkage map basically is something that you're going to draw a chromosome, and then you'll put the points in where the genes are linked by. Let's say this is big A and this is big B. If they're linked, you're going to write them down as, on the same chromosome, and you're going to infer that their centimorgan length is less than 0.5, and then that'll be if linked, or excuse me, it wouldn't be 0.5, it would be less than 50. If their distance in centimorgans is actually less than 50, then therefore those two genes are basically linked. If, however, you have a situation where you have a centimorgan range that is greater than 50, then therefore those two genes aren't linked and they will be exhibiting independent assortment. So you would have independent assortment. And then therefore they're not linked. If they're greater than 50 map units apart, you can't actually determine where if those genes are actually on the same chromosome, because remember you have multiple chromosomes involved in the situation. So it may be possible that if you have a recombination frequency that is greater than 0.5 or equal to 0.5, it could be that the different genes are located on different chromosomes, okay? So the recombination frequency is, like I said, a measurement of the frequency of how much you're getting recombinant gametes, basically. So the measurement of recombinant gamete and during gamete formation. So because of this, the recombination frequency is essentially equal to the total number of gametes divided by you're gonna use the denominator as total number of gametes, and then on the top, on the numerator, where you're gonna write down is you're gonna use the recombinant gamete, the number of recombinant gametes. So that's all recombination frequency is. Basically, what you're going to look for is you're going to look for the recombinant gametes. You're going to divide it by the total number of all gametes. And typically, the way how you do these, how you actually determine these, is you're going to use something called a 3-point test cross. We've already talked about what a test cross was way back in the beginning, but essentially what you're doing here is a simple, just basically a test cross that has 3 different genes involved. So what you're doing here is, we can simplify by just doing a single test cross, big A, big A. If you have an unknown genotype on the left and you're crossing it with an all recessive individual, this will be something called a test cross. And basically what this cross allows you to do is, first of all, it can tell you what the original genotype of this individual was, because we can either cross big A or big A little a, and we're going to get 2 different phenotypic proportions in the offspring by using that cross, right? This one is going to give you a half proportion of big A whatever and half proportion of little a. And this cross over here is going to give you all offspring are going to be big A. Another thing that these crosses allow you to do is actually measure the amount of gamete production for each of the genes. So basically what you can do here is that we can measure what is the probability of you getting this gamete being infused with an all recessive individual. So all these individuals are giving a big A, little a. So we know that we can actually use this method to measure the gamete production for each of these individuals from a test cross, right? Same thing over here. We can measure that in this situation we're getting a half. You have a half chance from parent number 1, which is this one, to get big A, and you have a half chance over here to get a little a from the first parent as well. So basically these types of crosses will allow you to do is measure the amount of gamete production by just looking at the phenotypes themselves, okay? So basically that's all it is. And we're going to do an example problem over here as well. Now, in this situation, you're given the gametes already, but 9 times out of 10, what you're going to see on an exam is you're gonna see their phenotypes written out, not something that was laid out easily like this, where they just give you the gametes and then you just have to calculate the recombination frequency. In an actual case, you're going to be given a couple of phenotypes, and basically you're going to use this test cross, and whatever phenotypes you have in the F1, if in the F2 generation, when you use a test cross, basically you're getting different proportions, and you can use that to figure out what gametes you had originally, right? Basically what I meant is that, let's say this guy is tall and this is short. Basically you already know that from this original parent over here, from this parent over here, they have to have given half of their big A gametes to make the tall phenotype, and half of their little a gametes to make the short phenotype. So basically, look at the phenotypes, what they're giving you, and basically that's correlated to the original gamete production that you had before. Okay. But this is just a simple question for you to just understand how to use recombination frequency problems. So to begin with, recombination frequency equations, you can only use it to compare 2 genes at a time. So what we're going to do is we're going to compare the recombination frequency of big A and big B. And I've already calculated the total over here, which is 782. So in these problems, what's critical for you to understand is the original parental genotypes, which I've labeled them over here. The reason why this is critical is because in the F1, their chromosomes the F1 generation basically has 2 chromosomes, one from both of the parents, right? One of their gametes is going to be, or chromosome is going to be all dominant genes, and then the other chromosome is going to contain all recessive genes. Right, because this original parent gave, and we're going to call that the dad. So this original parent gave this chromosome to the F1, and then the mom gave this chromosome to the F1 as well. So basically, over here, we have the parental gametes here, but what we want to do is we want to look for the recombinant gametes, right? So our parental gametes were either you have big A, big B, and then you have another one which is little a and little b. So to look for the recombinant gametes is essentially just a mix. So big A, little b, and then little a, big B. So looking for this, what you want to look for is a recombinant gametes. So something like, so you're looking for this individual, this one, and this one, and you're going to add those all together to figure out what the recombination frequency between A and B is, right? Because we're only focusing on A and B for now. So it's essentially 7 plus 6 plus 9 plus 7. Let me calculate that real quick. You get a number that is 0.037. Okay, and then what you want to do is you want to figure out the recombination frequency between every possibility of genes that you have. So now what we need to do is we need to figure out between A and C. So same thing, same exact mechanism. What we're looking for is either we're looking for the recombinants big A, little c, or little a and big C. So to find that one, so we're looking for big A, little c, or little a, big C. So one right there, one right there, one right there, and then one right there. So what we have here is 185 plus 190 plus 9 plus 7 and we get a recombination frequency that is equal to 0.5. Cool. Like I said, we want to do every single possible combination, so between B and C. Okay, so between B and C, we're looking for big B, little c, or little b, big C. So we're looking for those ones right there. So 185, 190, 7 plus 6, and then that should be pretty close to 0.5, but we can make sure, like 0.49, something like that. 0.496. So now what's happening here is that both of these recombination frequencies between B and C and A and C are pretty much equal to 0.5. So therefore gene C is not linked to either gene A or gene B. Okay, because the recombination frequency is equal to 0.5, whereas A and B are linked because their recombination frequency is equal to 0.037. So it's less than 0.5. So less than 0.5 means that you have linkage. So basically, how do we make a recombination map now, or a linkage map? So To figure out what centimorgans we have, we're going to do 0.037 times 100. You're going to get 3.7 centimorgans. So to create a linkage map, so this would be 3.7 centimorgans. And then essentially, because we don't know if C because C has a distance of 0.5 or 50 map units apart, we don't actually know if it's actually 50 map units apart that C is located between A and B. Because all these calculations they aren't measuring actual distances between different genes on our chromosome. They're just actually measuring the frequencies of recombination. So because of this, we can't actually determine if gene C is actually located on the same chromosome or if it's located on a different chromosome. You need more information for that. So basically, you wouldn't include gene C in this cross or in the linkage map. But that's basically it. That's essentially how you figure out the recombination frequencies. It's pretty simple. Always make sure to read your question. Make sure you figure out what the parental gametes are, and then you should be golden. That's it. See you all later.